This conference will now be recorded.
The program that she ran was the Human Microbiome Project, and I was just sort of in awe of how she could keep it all straight and organized and moving forward and under control. And she was a big inspiration for me in being able to think flexibly about how you work with large groups of people. So without further ado, I'd like to introduce Lita, who has come out of retirement to talk to this group about the Microbiome Project, as well as a lot of other things that she's doing in retirement. So, hi, Lita. Go ahead.
Thanks, Jen. Thank you so much. That's a very kind introduction. I'm going to respond to some degree with what you said, Jen. I think all I did is put the framework in place, and the investigators basically ran the program. They were all enthusiastic and all facing in the same direction towards a common goal. So maybe that's the larger message here. I understood my charge to be to discuss how we built the Microbiome Consortium through the HMP program. And what I want to do is highlight, there are lots of different ways that we built research communities, but I want to highlight sort of, I would call it 4 levels or 4 kinds of ways that we build community. And I'm hoping that some of these ideas are useful to the group that's listening in. So am I being heard loud and clear?
Loud and clear, yeah.
Yes.
Yeah.
Yes.
Okay, great, thanks.
Yes.
So
Yes.
what I want to do is sort of go over through about 8 or 9 slides, the NIH Human Microbiome Project, and we had a dual charge to build a research toolbox as well as to build the community. And I want to give you examples of how we did that over the years. And I hit the button. You know, I was testing this earlier. Oh, here we go. Okay, actually, I'll hit it twice. Slide 2. And my understanding is that we're going to share all of our slides. So this is a 10-year program, started back in fiscal year 2007, but of course the planning for it happened many years before. And by the completion of the program, we were funded at a level of $215 million over 10 years to build out this community resource. And many of the features, especially the early features of the program, took their lead from the Human Genome Project, because from the beginning of the Human Genome Project, there was an effort to build community, build consortia at the regional, national, and international level. So for the HMP then, our charge was to develop research resources, including things such as reference data sets, clinical and analytical methods, statistical and computational tools and pipelines. Also, to rapidly release these resources, so quickly and rapidly after QC, into public repositories and community databases. We had our own HMP Data Analysis and Coordination Center, which I'll explain in a minute why we needed it and why it was essential to the success of the program, GitHub, and other online resources, as well as meetings and webinars. And of course, by default and by design, both, we built a research community. So on the left there is this sort of logo of logos of all the different institutions and medical schools and so on, universities, that were a part of the HMP at one time or another. Let's see if we can get these little slides to move along here. Move along. There we go. The HMP had 2 phases. Phase 1 was really asking the question, who's there? Maybe more scientifically, to characterize microbiomes, number 1, through sequence analysis, through modern metagenomic sequence analysis, and to see whether we could correlate the microbiome composition with phenotypes. And we accomplished that through 2 large-scale efforts. One was the so-called the famous benchmark healthy cohort study, in which 300 adult men and women, mean age 26, were verified to be free of disease across 5 major body regions, skin, oral, nasal, urogenital, and GI, and then sampled across those 5 major body regions. We also had a second set of cohort studies, which we called the demonstration projects, to determine whether we could in fact demonstrate whether there was a characteristic microbiome associated with these 12 different clinical phenotypes. Now, we didn't have a particular interest in the phenotypes per se, because the larger question was to say, could we in fact predict host phenotype by microbiome composition alone? So we did things such as not only analyze microbiome composition, but also try to predict the metabolic pathways that these microbes, these metagenomes, appeared to code for. As you probably already know, with just a very casual reading of the literature, you cannot predict phenotypes with microbial composition alone. And I'll give you just a super basic example for those of you who may not fully appreciate this. You know, we have E. coli that lives in our GI tract. It's a normal member of our gut microbiome, but there's also E. coli that causes all manner of food poisoning. Those microbes are both named E. coli, but you cannot tell me which one causes food poisoning and which one, which one lives as a normal member of our gut. So we had to go on to actually look at the functional properties, the biological properties of the microbiome, and that comprised phase 2, or asking the question, what are they doing? So phase 1 ended formally, if you will, at about fiscal year 2012, when a series of flagship papers and companion papers came out in Nature and sister journals of Nature in 2012. And then the year thereafter started, of course, phase 2. This was a much larger undertaking, believe it or not, because of the vast array, as I'm showing you here in this cartoon, vast array of functional molecular properties of both host and microbiome that were collected. We collected these properties over time, so these were longitudinal cohorts as opposed to phase 1, which were case and control cohorts, and we really had to limit ourselves to 3 conditions: type 2 diabetes, inflammatory bowel disease, and preterm birth. We weren't necessarily interested in those conditions only, but the goal was to collect these suite of properties in host and microbiome over time. And that way, anybody who wanted to ask the question, which of these functional properties provide the most insights, we gave them 3 case examples. And of course, all of the data, the clinical protocols, the pipelines, and so on were all deposited in our HMP DAC. And the reason is, you know this from your own work, we don't yet have repositories that are federated or linked, and it's almost impossible to reconstruct a cohort study and all of its parameters and so on by just going to the public repository. So we knew from day one we needed to have our own repository that allowed an investigator to go in and look at the data, look at the studies that were conducted, and understand what properties were collected and under what conditions. So next slide, then, slide 4 is sort of a cartoon again of how much data is in the HMP research toolbox. Phase 1 is a total of about 10 terabytes of data. Really limited the sequence data. Phase 2, although much smaller, was double the amount of data at roughly 20 terabytes of data, and that included multi-omic data. So this is just a cartoon of how the data look. The colors code by either host data, microbiome data, or mixed data coming from both host and microbiome. And the size of the dots are meant to tell you how much data was collected under that category. The first 2 columns tell you the data that came from phase 1. The third, fourth, and fifth columns tell you the data that came from phase 2. And that's the website if you have an interest in going in there and perusing what was collected under what conditions and what you could learn with those data. The publications for phase 2 were published in 2019, again in Nature, a series of 3 flagship papers and a series of 30, I think it was, 30 or 35 companion papers that came out of phase 2. So I said we wanted to create a community. We certainly created a community within the HMP. The other question was, did our toolbox do any good? And in fact, we decided at the end of HMP, we were going to sit down and peruse through the NIH grant database and figure out of all the microbiome studies, how many actually made use of the HMP resource. So this is over 10 fiscal years, the same 10 fiscal years that HMP was active. I want to draw your attention to the red and blue bars on this graph. The red is all of the annual disbursements for HMP. More importantly, the blue are all the annual disbursements across the NIH for non-HMP funded investigators. These folks had nothing to do with the actual program, but got interested in or got engaged in microbiome research. And I want to point out a few things to you. First of all, it's $880 million over 10 years. So that's 4 times what the HMP was funded at. Perhaps more importantly than the numbers, the dollar signs was 21 of 27 NIH institutes and centers now fund, possibly more now, but back then in 2016, we calculated 21 of 27 NIH institutes and centers to fund microbiome research, whereas in the beginning it was, I think, 4. And this data is a summary of 1,000 plus studies, and we read every single abstract and special aim, and these 1,000 plus studies, in fact, did cite the use of HMP reference data, protocols, and analytical pipelines. So we feel like we catalyzed and, in fact, democratized human microbiome research at NIH as a result of the outputs from this program. If you're interested in more, and I got to say, Rolanda, I don't know if that turquoise horizontal line, does that cut off the bottom of my slide?
No, it doesn't. You can see that.
Okay, great, cool. You'll see there's a reference here. We did quite a deep dive into the grant portfolio at NIH, and there's a lot more rich information about the human microbiome portfolio at NIH over this 10-year period in this publication that we put out last year. And of course, NIH being NIH, most of the attention was on diseases, but if you see in the lower left-hand corner here, you'll see that over 5 years, we did a deeper dive over the last 5 years, fiscal year 12 through 16, about 1/3 of the total investment was in actually basic biology of the microbiome. That's our sort of general category. 2/3 of that investment, that $728 million over 5 years, was into diseases. And in fact, we used the WHO's ICD-10 classes of disease to try to categorize the different diseases that were being studied. And we estimated over 100 ICD classes of disease are being studied at the NIH. And of course, this is just a cartoon of the various categories of disease and body regions in which the microbiome plays a role in disease. So I mentioned that phase 1 ended in fiscal year 12. Sometime around that time, as things were rolling along, I began to realize that all these different institutes and centers at NIH were funding microbiome work, but we really weren't talking to each other. So I decided to establish a Trans-Nih microbiome working group. So that's sort of level 2, if you will, of building the community. It is extramural program staff only, and membership quickly came in from these 20 or 21 different institutes and centers. I did start the program. Now we have Ryan Rinaldo at NIAID and Phil Dashner at NCI, who have very kindly agreed to continue the TMWG and serve as the current co-chairs. And you can see the mission here. It really was meant to be a forum for us to discuss what was happening in the microbiome area at NIH, everything from creating workshops to publishing that paper I mentioned in 2019, talking to each other about upcoming funding opportunity announcements, basically a clearinghouse for us to discuss with each other what we were doing in this area. It also meant served double duty because it served as a central resource for the external community. I just tapped in the current website for the TMWG, and the reason is, the NIH is quite a large and gargantuan and complex agency, and this young research community was having difficulty figuring out where to go. So this was a way for a new investigator in the area to be able to figure out which institute was funding microbiome work, in what area of microbiome work, and then each one of these program directors agreed to be their point of contact for their institute in order to direct and assist any investigator who may want to explore microbiome research funding. So that's sort of one level of community. Another level of community that actually has just recently come to pass is, of course, community is built through, research meetings and conferences and workshops and publishing together and doing research together. But in fact, this better than I, in fact, the microbiome field is a multidisciplinary field. It involves so many different expertise to come together to do the work that I think that, I don't know, we're still in the early developmental phases of the field, but there is some signs of maturity showing up, because last year there was a workshop called at UC Irvine last summer in which 36 microbiome centers, and let me explain about these microbiome centers. You'll see this map of the U.S., and you'll see these red stars all over. These are basically grassroots, mind you, microbiome centers formed because a dean or a provost or someone gave these folks seed money to kickstart a community on their campus. Okay, not all of them are focused on human. In fact, maybe half of them are, and that's actually an important feature. After that Irvine workshop, there was an agreement. There should be a microbiome centers consortium. So now we're talking about community at the campus level to now community at the national level. There is now a microbiome centers consortium website, and I serve as an advisor to this group, and a publication was put out, a perspective in Nature Microbiology. I feel like it was either December of last year or January of this year, basically announcing why we need microbiome centers and inviting others to join. So that's sort of the next level of community that has formed over the last, let's say, 10 to 15 years of this field. I went through that pretty quickly because I think my time is fairly limited, but those are just 4 kinds of examples of how community was built and all the major benefits that came out of building out communities at these different levels. So I can stop there, Jen and Rolanda.
Sure, we can take questions. Everyone is welcome to type the questions in the chat box, or if you'd like to speak, I'm sure you can do that too. I think we can take about 3 minutes for questions, yeah? And then we'll give the opportunity to the next speaker.
Okay, let me let go of this slide. Where is it? Here, stop sharing. Okay, I'm putting myself on mute.
Sure.
So Lita, I'll start us off with a question, which is, I think you gave a nice overview of sort of how much the field and the community changed from start to finish. And what's your sort of sense about integrating with data that was collected at the beginning, and does it need to be sort of recollected with current standards and
Yeah, it's an excellent question, Jen. In fact, there was a lot of discussion about that because for phase 1 especially, there was very deep sequencing done of the metagenomes and the 16S data, but particularly the metagenomes. And in fact, it turns out that the community standard is not as deep. I don't remember if it's 30x and now the community standard is 5x. I don't remember, but it was something pretty radically different. So I was told by scientists in lots of meetings that they had to actually refilter the HMP reference data sets to remove that depth of sequence because it wasn't going to match up with other reference data sets they wanted to compare against. And we also had the same kind of conversation about redoing the data with new standards because, in fact, my God, during this whole period, as you best know, I mean, the technology was radically changing in ways that are, frankly, mind-boggling. So there are sequence technologies now that no one could have dreamed of 10 years ago. And so I think, in fact, you're right. probably the community will come together and realize we need to redo this, either by samples that have been biobanked, so you could resequence biobank samples from a former cohort, or you have to start with fresh cohorts and fresh technology.
So we have a question from Scott Hazelhurst. What were the challenges in getting started and lessons for building communities in other parts of the world? And specifically, what are priorities if you have limited budgets?
Yeah. So Scott, because it was a mandate of the HMP, we had to build community. So we built in many mechanisms for trying to reach out to the larger research community. That was easily done, obviously, because there was a lot of interest in what the microbiome could tell us about human health. And so there was a huge interest of people joining. You know, I'll give you the example of the microbiome centers, the map I have of the U.S. with all the microbiome centers. Based on what I learned from those 36 early founding centers, those were almost spontaneous. It involved very, very little money. It involved the recognition that no one lab could do it on their own, that either they had the microbiome expertise, or they had the computational expertise, or they had the ecological expertise, or whatever, but they didn't have it all. So this might sound a little naive, but frankly, even though I know this was a big budget, some of that community organization happened almost spontaneously through need and desire.
All right, thank you so much, Lita. We really appreciate it. And I'm sure that if people have questions later, they can send them to Rolanda and she can perhaps get some additional written answers.
Yeah. And if you don't mind, I'd like to sit in on the other two talks.
Oh, please do. Absolutely.
Thank you. Thank you.
And so that brings us to the next talk. Lynn Shermill, actually, I have known for a very long time because we were postdocs together in the same lab at the National Cancer Institute. And she has gone on to develop a strong research program at the University of Maryland and was part of the Human Microbiome Consortium and continues to work in the area of standards. And I'll also say our daughters have been friends since kindergarten. So we've known each other a long time. Lynn, do you want to take control of and share your slides? There we go. I can see them.
Sharing screen. Hopefully everyone can see it all right. I'll make it bigger. Yeah. See. There we go. Can everyone see all right?
Yeah, it looks great.
All right. Okay, great. All right. Thank you, Jen, for the invitation. Lita, it was a great talk. So I'll again, same theme. I'll talk about building communities. So this is a little bit different in that this is a grassroots building of a community. So I'm going to talk about genomic standards. Not the laboratory like positive control standards we were just talking about in the questions, but more on the metadata standards, the context of the sequencing. So I want to walk you back about 15 years ago. We're thinking about when the human genome was done, microbial genomes were ramping up. Many people in the community were coming together in consensus and saying, we have no way, or really great way at that time, to compare our data. So we were thinking about studies, environmental studies to begin with, soil and water, and hundreds of samples at that time. It seemed like a lot, right? It's small potatoes now, but it was a lot of data, and we didn't really have a great way that we could conceive to standardize what we were collecting, so we could compare studies over time across studies, and that was really the impetus for us coming together. And today I'll go through what we've done, how we've grown, what the consortium consists of, and then hopefully make some mention of how it could be reused in this project, and I'll mention how it's being reused in many other projects. Okay, great. Okay, so I think the heart of the Genomic Standards Consortium is our community. What I'm showing you photos of here is our current board members. We have another 20 or so retired board members. We've been together 15 years. As a community, we first came together with the understanding that we needed to find a way to work together no matter what we studied. So on this page, you'll see virologists, biodiversity researchers, and people interested in plants, the full swath of researchers, but also you're seeing people that are involved in large-scale data sets, data repositories, journals, the full gamut of research you can imagine in genomics. So over time, and I've got a number of their logos here to show you the kind of the breadth of where they come from our current board. So for the last 15 years, we've come together in the early days twice a year because we were building standards. We wanted to have consensus on the sets of data types that we would collect, and I'll show you what we've done with that. Over the years, our mission has evolved from just building standards for the data we were needing, and that's showing you all those various diamonds on the map in the back end. We have had people join our consortium. I'd say we're about 500 people on and off over the years. We move every year to a different location for our annual meetings, and that is an outreach initiative. So we can talk to postdocs and students in those areas and different researchers. But we also have thematic meetings where we're trying to tackle where are standards needed in the community at a particular time, or where do community members need to come together to discuss their standards. So that's really where our mission has evolved to. But a big aspect of putting standards together is getting them used. So in the earliest days, we needed a standard. We worked on that. We quickly came to the realization that if it's not implemented broadly or not utilized by people outside of our community, it's not as of great a utility as it could be. So the HMP is a good example of that. So our consortium started maybe 4 or 5 years before that. And when the HMP was building at HMP DAC, Owen White, who was leader of HMP DAC, he was part of the consortium very actively now. He's on our advisory board now. Anyway, so when data types need to be developed, we were in, fortunately, a position to help that community as well grow with the context of the data being collected. So I really want to say one more thing about this picture is that it is really the efforts over many years of people putting in their time to realize how important it is to collect the data that we're comparing, right? And so that's the strength of the consortium. We are as I said, grassroots consortium. We've had some institutional funding here and there over the years, but majority is we all just contribute. And we are fortunate to have some corporate sponsors now for some of our annual meetings, but by and large, it's just a community effort. So what we're trying to collect is the context of what we were sampling and then sequencing. And this slide sort of shows you the components of what we needed to think about. So we had samples from different environments, we had different sequencing technologies, and as we all know now, even the technology can impact what the sequence is on the other end when you're doing analysis. So figuring out what was going to impact what we were going to see, what was critical to collect for particular communities, that was important as well. And that's part of our evolution. But at the same time, having people in this blue cylinder you see on the slide that were putting together or part of these very large data efforts, that they were part of the consortium. It's critical that they be part of it, the journals be part of it to promote standards and utilization, but we have to have standardized tools and infrastructure so that the standard is a useful tool. The INSDC on the far right, they have been part of the consortium since the very beginning, and that again is a critical component, that all the people that are like the key aspects of where the data will be, how it will be represented, how will we reach out to communities. So this is kind of the bigger picture of where we kind of fit in the middle of all of this. This is our homepage for the GSC Consortium. So we formalized over the years, maybe year 4 or 5, we formalized, we have a board, we have our annual meetings, as I mentioned, but we also have a mechanism to reach out to community members to actually identify through these members, through our own community and others, what areas of standards need to be developed so that we're an open society. Anyone who wants to join can join, there's no cost. People say they have a project, they want some help, we're more than happy to do that, and that's kind of how we've continued over time. We are very multidisciplinary, as was mentioned earlier. It's a critical component, I think, to these kind of large-scale efforts because there's many different needs within a consortium, but within what needs to be developed, outreach activities, tool development, so different skill sets, but then they all kind of come together under a common need. So over the years is continued that we have a common goal throughout our group, our mission, to get this data discoverable over time across studies and to continue that effort. This is just a page describing what the standard is that we developed. It was critical in the early days to identify what key terms could be utilized across any kinds of studies, because at those days we were looking at like the English Channel studies, soil studies from in Michigan State, but then foreseeing human microbiome was about to happen, and we knew other studies would be coming. So in the box on the left side, these are the types of data we thought everyone could collect across whatever study they had. We wanted data to be as rich as possible, but also to have it in this kind of component system, because then we could expand. So like say when we were studying the built environment, so one of the products I work on, buildings, cars, subways, trying to understand the rooms you're all sitting in right now, how the microbial community is impacted by the space that we're in, by the humidity, the airflow, do you have an open window next to you, those kinds of things. So we wanted that expandability of possibilities for comparison, at the same time having a core bit of data that we could all compare. To date, we have 20 of these packages published and in use, and I'll go over some of those in a moment. It continues to grow. We'll continue to work with communities, but again, we want it to be flexible and to be able to be extended as needs arose. So some of the outcomes here, this is a slide I'm showing you outcomes on the left side, the benefits of building a community in this structure. One aspect of it is as communities grow and have needs or are developing, or maybe they get very mature and then other communities arise. So this is really what the diversity of packages, we call them packages, they're really lists of terms. They arise as the data is needed in the community. As different types of data have grown, we work with new communities. And again, on the left, the benefits. So I was checking again just this morning in the biosample database, for example, at NCBI, we have over 700,000 records now with our standard. In the early days, it was all of us, our community, we were like 20, 25 of us at the beginning, but it has grown, not exponentially, but quite nicely. But with the idea that we wanted data to be discoverable. We were doing, everyone talks about FAIR these days, the findable, accessible, and all. That was a backbone of what we were doing from the very early days. One critical component is, yes, at the beginning, we were the people in the consortium for the most part doing soil and water and the HMP, and so a lot of experts in the room. But as we've grown over time, clearly that was not a model that works. So we've always outreached to other communities, and the community itself determines the standard. We are just the helpers, really. We help guide it. We have a pipeline with the INSDC. We help coordinate, and we help encourage strongly people not to use free text, to reuse terms that are already in the standard. So best practices really is a lot of what we push forward to help people kind of have their data be as comparable as possible over time. And it really has helped a lot. And I'm sure as many people on the call will have been at seminars and heard comparative studies to the HMP. Well, those comparative studies are based much on that the data was standardized. It was rigorously collected and recorded, and then people could do this comparison. It's a great example of a win of having a large, amazing data sets, but also at the same time having it very standardized. So just going down the list a little bit on the right, you'll see that we have newly published packages near the bottom, including uncultured viruses. We have new communities we continue to work with that publish their standards or evolving their standards. And then you see some of this here if you're going to the INSCC. This is a NCBI page. It shows a diversity of options for types of studies that people are doing, but there's also a launching point. So say if you had areas of work you wanted to collect, and one of these packages didn't quite fit what someone's doing, we have the ability to work with groups and help kind of repackage them. So it works better for their community and the bits of data that they want to collect. One of the other type of win or outcome you get from doing this kind of community development is the tool development. So this is an NCBI biosample page, and you'll see us saying similarly, a little different, but similarly over in the UK at the EBI, as that you have this tool development on the other side of the work, where you're able to richly research into the data itself. So it's not just a text search like Google or anything, but you can actually have detailed collection and association of bits of data that you can then compare and mine. And I'm showing you on the left side of the slide. I actually pulled out this example for a class I was teaching recently. You can get very deeply into the data because it is tagged, it is regularized. So that's actually another win of it, is having this very rich ability to do comparative analysis. And that's near the end here. I'm showing you some of our publications, some of the groups we're working with. We do encourage groups to reach out to very large communities when they're building a standard, because it should be inclusive. We don't want, we don't actively encourage people to be just like my little group and maybe the lab across the hall. No, it is a community effort. And I think if you're going to have success in community-driven standards, you need to have buy-in, and you have to hear the community members. So one really good example of this is the viral one you see on the left side. I'll show you a slide in a moment of all the participants from these co-authors, but it's a very vast community that came together, very multidisciplinary. And just one last point on this slide, showing you some of the groups we're working with now that are building data sets, have reached out to us, and we're coordinating and helping them put a standard together. And I think one of the benefits of the outcomes of what we've been doing is, you know, we're an open community, so we're just helping these groups bring their data together. But one of the nice advantages is that with the INSCC, the pipelines are all in place. So if people build a new standard, like the viral group did, a large product like that, we can get it in place within about a month once the standard is developed. So the INSCC is a fantastic partner in all of this. And so people don't have to reinvent the wheel, and they can compare their data. Maybe the FDA's data, you know, in food or pathogens, they may want to look at some of the other packages where data has been collected previously, HMP maybe, or some other, and they can, because the commonality of the same terms used to describe the same elements. Just one, I think this is just about the last slide. So on this point, showing you that data is collected, and then you can see how it was represented here. But when people are building new packages, they can pick from the various packages what components, maybe for their studies, they want to decide their community should absolutely report every time, or whether other optional studies. So there's quite a large variety of standards there already for reuse, but new terms do also get created. We put a new standard out about once a year, because it has quite a long vetting process it has to go through. And this is just a picture to show you it really is community-based. These are all the people that were involved in putting together this uncultured viral genome paper and standard, and that then you have the community utilizing it as well. So I think that's a critical component of it. And then just the very last slide, like I mentioned, our annual meetings. We are postponing our one for this summer, obviously, but we will be hosting it hopefully in the late fall or early winter of this next year. So just keep that in mind. It is a great opportunity to discuss standards, learn about them, and it's open, and we'll probably be back in the States the year after. I think we're going to be at Berkeley. So thank you for your time. If you have any questions, let me know, or contact me later. And that's it.
Thank you, Lynn. So once again, I'll start off with questions that, since you're on this slide. I noticed on your original map, there aren't a lot of participants from Africa. And so one is, would you want to do some outreach in that area? And then the second is, would you consider holding a meeting in Africa in the future?
Oh, yes and yes. We are very minimal. As you get, sorry, I stopped screen sharing. I didn't know if you needed it, but yes, we have had people from different parts of Africa join us at meetings, and we would be more than happy to host a meeting in Africa and have people come to our meetings. We also have a monthly working group, which is an online activity where we also invite people. We can do the work, some of the work ahead of time, or help people deal with their standards challenges. But yes, absolutely. I think the reason we're still going after 15 years is because we've been so open and wanting to work with new communities.
All right, so now it's up to this community to reach out to Lynn. And again, you can always write to me or Rolanda to get in touch with Lynn. Any more questions? Great, Rolanda, are you going to introduce Voki?
Sure, Jean. Um, well, I'm sure most of you have read Voki's very nice bio sketch. I know Voki as an H3Africa fellow. She's based at Wits University and working with H3Africa PI Michelle Ramsey. Okay, over to you.
Yeah, hi, thanks, Rolanda. And thanks to the two awesome speakers that have really given us some insight into the field of microbiome research globally. I'm also quite thankful for the opportunity to present on this platform. I hope in a few years I can come back and say, 15 years ago, we started doing such and such, and now we are here. But for today, I've put together a few slides just to give some perspective on the trends of microbiome research on the continent that's in Africa. I've also drawn from a review article that was published with our collaborators about a year ago. And so this talk is divided into 4 short sections. I'll go through them very quickly. Just pretty much microbiome research. Where do we stand in Africa? And sorry, I have to go back real quickly now. The current research focus in terms of sequencing technologies, body size, and also the problem with the research in Africa, and also the implications of this gap we see in microbiome research compared to what's going on globally, and just a few thoughts on the way forward. So where do we stand in Africa? Microbiome research. There have been more than 9,000 studies published around the world to date. And Ryan, one of our collaborators from Stanford, put up this slide in a presentation he did about a year ago. I've modified it slightly just to give a perspective of what's been done globally and what's been done in Africa. This is a question I asked everybody, and just to have a sense of what we were thinking. And what do you know? We actually are at less than 100. So out of the more than 9,000 studies published globally, there are less than 100 publications with African participants. That's a rather incredible number in the larger scheme of things, but that's actually where we stand in Africa. So I believe this gives some kind of substance to general thinking that African gods are clearly underrepresented in microbiome studies globally. Less than 100 studies equates to about 1.2%, spread across different body sites, as we can see, with the majority being the gut microbiome. We find that less than 15 of these studies has been on chronic diseases. This number really does of the health because of the gradual shift that we know health burden on the continent towards NCDs rather than infectious diseases. And I believe the WHO highlighted that NCDs will become, excuse me, the leading causes of morbidity and mortality in Africa by the year 2050. Also, quite interesting is that about 60% of African countries there's been no studies done on about 60% of African countries, which pretty much just tells us that there's so much more to be explored with respect to the microbiome, be it a gut or any other body site on the African continent. So what has the focus of some of these published studies been? I have to say though that there's been quite a flurry of research this year, published studies, I should say this year. The keyword here is published studies. So these are approximate numbers, but it gives us a general idea of where we stand. So the focus of this published research have actually been on the traditional microbiome, the H3Africa, which is very important, comparative studies between Western African populations, very insightful, the coevolution of the microbiome, that's how the microbiome has evolved with its host, the development of the microbiome from infancy, its role in infectious diseases, the role of the gut microbiome on vaccine response, and in ready-to-use therapeutic food supplements in case of malnutrition, which is also a major challenge on the continent. There's also been studies in cancer and mental health. And just some general notes on these published studies. I think it's important to note that most of these studies have actually focused on, or have actually been done using the targeted amplicon sequencing, which is ribosomal RNA gene sequencing, which is understandable, as this is generally affordable and broadly informative. But like Lita said, I think this has to do, I think most of the majority of African studies are still on the who's there phase of microbiome research, and this is what this does pretty much. It pretty much gives you, it's informative to an extent, but it primarily provides quantitative descriptions of microbial compositions. It's constrained to bacterial and archaeal classifications, and as such does not really allow for adequate harnessing of the full information from the samples that are available. I also thought, just bearing in mind there have been some published studies, but then it will also be good to point out some work in progress that actually caught my eye since we're looking at the trends in microbiome research on the continent. Just a few studies I came across from the H3Africa meeting that was cancelled in Kenya, the agenda, and also some of them actually presented in the online format. And these are some of them. The breast milk microbiota influence in infant immunity and growth. I found very interesting, and it's headed by Alashia Bimiku, Jonathan Blackburn, and Clive Gray. I'll refer to this study just before I close my presentation. And also a study on cancer, integrated biorepository of H3Africa, Uganda, investigating the gut microbiome profiles associated with programmed cell death amongst men with prostate cancer in Uganda. Then, of course, there is the AWIGEN project. There's a microbiome component to the AWIGEN project. AWIGEN generally focuses on the interplay between genetic, epigenetic, and environmental risk factors for obesity and its associated cardiometabolic disease in sub-Saharan Africa. So there's a microbiome component to that as well, which has been headed by Prof. Hazelhurst, and it's actually a very interesting study going on as well with our collaborators. Then I thought this is pretty a pretty neat project from the H3 Bionet, headed by Dr. Usema at the Pasteur Institute in Tunis. I'm going to just quickly show us what it looks like. It's actually a portal of, the general idea of the portal is to provide like a one-stop location for all African-related metadata. It is not yet live, and will still very likely undergo a few aesthetic changes and tweaks, but should hopefully be available in the coming months. And I think it's definitely a resource to look out for. It's just, this resource is like trying to bring out, what's been done at one point, so we don't have to go searching for African studies across several databases, and it's curated as well. So, and just some studies that were quite a delight to see this year. The first study profiling the gut microbiome with type 2 diabetes from Charles Rotimi's group, and we also have a study currently under review of the AWIGEN project as well, with our collaborators from Stanford, and it's titled The Gut Microbiome Profiling of a Rural and Urban South African Cohort, reveals biomarkers of a population in lifestyle transition, which is actually very, very critical in terms of where Africa stands now. I'll elaborate on this in a few slides down the road. Just talking about the research gap and its implications. So now I did talk about the transitional state of African research and the fact that some of the focus of African microbiome studies have been on very traditional populations, right? So the thing is that we end up with an overapplication of these studies, the current hunter-gatherer studies, which in fact is a relatively small population, a small group on the continent, at the expense of understanding populations transitioning or recently transitioned into more Western lifestyle practices on the continent, which is really the state of many African countries. They are transitional populations. Not to take anything from the traditional studies, they have been very crucial and really critical to our understanding of the microbiome. But then this problem is also compounded by the fact that the current studies going on as well have used, like I said, 16S sequencing methods, which generally leads to an underestimation of the gut microbiome diversity in African populations and very likely restricts the ability to comprehensively understand and act on key medical and scientific issues. So just the last two slides here, just drawn some thoughts from the paper I talked about earlier, the review, highlighting some outstanding questions that we're looking at and some opinions and some concluding remarks going forward. I just put a few things there highlighted because of the shift from infectious diseases to more NCDs on the African continent, and it's very critical. It's important for us to know what's going on in that regard as it affects the African microbiome. HIV is a problem. It's also how does the microbiome, HIV medications influence the gut microbiome and infectious risk? And also something I felt was very crucial, especially in this in the current state of the world, to what extent can the gut microbiome explain differential vaccine responsiveness between Western and African populations? So these are all things that make African research on microbiome, on the African microbiome generally, very crucial, because for the reference databases to be better represented, to be better, and to provide more information, it has to be better, African guts have to be better represented on these databases. So just a few concluding remarks. I felt that I'll share some of my opinions based on my little understanding so far, my few years in microbiome research. The thing is that we may have come to the party a bit late in terms of microbiome research, but I think there are some advantages to this. We can learn from some of the lessons, I believe, from global research. In terms of standardization, I think Lita did mention this in response to Jen's question earlier. I think since we are just kind of in the beginning phases of microbiome research in Africa, one thing that is worth paying attention to is standardized setting protocols. We all know that good samples are the basis of good research, and just standardizing some sample collection processes as well as sequencing processes, because if at this point we're doing a lot of 16S sequencing, which is great and affordable for us right now, but at the same time in the future, we might want to invest in long read sequencing or shotgun sequencing that will provide more informative data on African guts. And we need to prepare for that. Like if we have standardized sampling processes and extraction processes, the sequences we extract now can be, or the samples the DNA we extract now can be used down the road, and we won't have to go do some recollection of samples because we might not have the opportunity to do that. But then we'll have them stored somewhere, hopefully, and be able to access them, and they'll still be of good quality for future sequencing technologies. And also just being able to have the standardized process as well to allow for meta-analysis in the future, data integration in the future, to be able to harness more information from our data with bigger samples. But then again, just going back to the fact that we need more diverse reference databases for better taxonomic profiling of African data, and this can only be accomplished by having proper good quality data and sequences. And then important again to the growth of microbiome research and proper research in Africa in terms of microbiome is to forge partnerships, international, intercontinental, transcontinental collaborations. I really enjoyed the part of the community engagement and the community building that Lita and Broadening talked about, and how crucial this has been to the success of microbiome research in the US and in other parts of the world. So just lastly, to conclude, I just want to acknowledge the collaborators on a review paper, which I drew quite a bit from, and H3's Awigyan project, the Sydney Brenner Institute for Molecular Bioscience, and my supervisors for giving me the opportunity to do what I do. Thank you.
Thanks, Voki. Are there any questions, Voki?
Hi, I do. It's Lita Proctor. That was a lovely talk. Thank you for that overview. I really appreciate it. So I think one thing that we didn't do very well in HMP and maybe in some of the early days was to really leverage ongoing existing studies of various diseases and making sure samples were biobanked for future study. So I know it doesn't address the need for work we need to do now, but if somehow African scientists could biobank samples in an organized fashion so that microbiome work could be done later as costs of sequencing drop and also as other types of analyses are more available, that would actually help quite a bit in the field. Do you know, Voka, what is being done within H3Africa or perhaps in other programs around trying to biobank samples for future microbiome analyses?
Thank you. I am not quite sure of the things in place at the moment, but we are trying to we're kind of trying to get started on collaborating across Africa to just get this going. There's a task force that is in its beginning phases, and I think this is one of the aims of the task force to figure out a way to collaborate and get these samples, very useful samples, biobanked somewhere. I think this is one of the aims, I believe. Jen, Scott?
Yes, if I could perhaps add to that, I think we are keen on doing some biobanking. We have been a bit reticent about it because when we, at least at WITS, when we started our project, when we did community engagement, there was concern about long-term storage of fecal samples because of beliefs in the community about how the material could be used, for example, for witchcraft. So we were perhaps oversensitive to this point, but in our initial projects, we said we would not. Once we'd extracted the DNA, we would no longer keep the material. And I think that is an issue that we need to revisit from a scientific point of view, but I think we do need to be sensitive to it.
Thank you.
Vuki, there's another question in the chat box from Mark Engel. He says, thanks for a great talk. For the ability to do meta-analysis later, do you have a template database which can be used and modified?
Thank you, Mark. We don't have anything at the moment, but these are just beginning stages. I believe this is something we're going to work on as well. That's a very good question. We're going to work on it, but we don't have anything at this moment.
So do Lynn and Lita perhaps have some insights of where you might find starting point?
I have a suggestion. This is Lynn. So the EBI, you may have noticed Rob Finn on the first early slide, or not. He leads the metagenomics analysis group. He's part of the GSC. He has a suite of metagenomics analysis tools. Magnify, MG, NIFY is one of them I would point you to. And they are a highly collaborative group, and I'm sure would be more than willing to help and to collaborate on tool development. That's one of the first suggestion I would go to. I think they're leading the world in doing that. There are many other groups that do it as well, but they're one of the groups I would suggest to start with.
Thank you. Noted.
Lita, what do you think?
Well, as I had mentioned earlier, all the tools and analytical pipelines are available on the HMP DAC. I caution you that because the HMP program has sunset, it has completed, we don't have active staff on board to help investigators, but at a minimum, you can peruse through the reference data sets and the tools that were used as a part of the HMP program, and that will still be useful, but you won't get the kind of support and interaction that Lynn just described.
I also put in the chat the Cheetah or Time database at UCSD. This is Rob Knight's group. He was one of our early members as well, but they're also quite collaborative. Both groups do courses and will come on site to local communities, research groups, to give courses on how the tools are used. But they're also, I think, quite interactive with the community on tool development.
Great suggestion.
Yeah, thank you.
Are there any other questions?
This is Jen. I just want to add that I think part of the reason to have Lita and Lynn come and talk and hear about what's going on in Africa is so that this moves forward sort of as an international effort, and that we're not recreating the wheel where we don't need to, but integrating and adapting things that already exist.
Yeah, I would second that, says Lynn. As Lita and Jen just said, there are many more tools out there, groups doing things like the Elixir from the EU. They have some very deep pockets. They also have a number of programs that they could that include international partners. And I know the Elixir is trying to have additional international partner hubs. So that, yeah, there's much. Back when we all started, we didn't have tools or resources out there, and so I think there's a lot you can take advantage of, and then reuse, and like you said, make partnerships for.
Hi folks, this is Ami Bhatt from Stanford. I have the pleasure of collaborating with Voki and Scott Hazelhurst and Zane Lombard. One thing that I just wanted to comment on that I find really impressive is the African efforts to overlay microbiome research on existing health and demographic surveillance studies. So the effort has been made to collect microbiome samples on individuals who have been very deeply characterized from a metadata perspective, and a lot of these subjects have been followed over time. So this is something that I think was really new and exciting to me, and something that I have definitely learned from my colleagues there. So I just wanted to give kudos to them, but also point it out as a really great way to make these data even more informative, because we know a lot about them.
I mean, that's a really good point, and I also think that you know, there is so much more to be learned, and it's a sort of rich, fertile ground right now, because as Voki pointed out, it has not been as explored as it could be at this point, and so there are a lot of opportunities. Michelle had a question about community engagement as key to broadening microbiome research in Africa.
Yes, thank you, Michelle. It's actually crucial because, like Scott mentioned, that the community we worked with needed to actually be sensitized to the fact that the stool samples we're collecting will not be for witchcraft, because you need to really engage with the community, talk to them so they have a sense in very, very simple language what you're working on. Because if that is not done, it's going to be a huge barrier, because I always feel like with African research, especially with the microbiome and the kind of samples we collect, that is the first victory. That's almost like half the problem solved, or half the research done really in Africa, if the community has an understanding and are willingly able to give it the samples you require. Sure.
Right, I think we've now gone over time. Are there any other pressing matters?
Thank you for inviting us to join your meeting today. Really appreciate it. It's really great to hear about all the progress you're doing and all the future plans. Very interesting.
Thank you
And
so
thank
much,
you very much
everybody.
to all three of our speakers.
Yeah, I really enjoyed it. Thank you.
Thank
Thank
you.
you all for
Bye-bye.
keeping on time. The overtime was because of the interest in the discussion. So thank you very much. We really appreciate it.
Sure, of course, of course. Thank you.
Thanks,
Thank you.
everyone. The presenters will make the emails available, so any further questions can be directed to them.
Oh, there's one more question from Laura Dugas. For those of us performing large microbiome cohort studies in African populations, is there an opportunity to get involved in the task force mentioned? And I believe the answer is going to be yes, but the question is how. So Laura, if you send a email to Rolanda, she will hook you up with the task force, I think.
Yes. Yes, the slides, the whole recording will be shared onto these mailing lists. So that will include the slides. All right, thank you everybody for your attention and for joining, and to the presenters. Some of them have already left.
Thank you.
Thanks, everyone.
Thank you.
As well, and Harry. Right, bye.
Thank you. Thank you.